Hi, in this video we are going to explore how to do enrichment analysis of antibody libraries obtained with display technologies. Here I already have a pre-set up project where I have 4 samples coming from 3 rounds of enrichment, and the key for the enrichment analysis is the proper metadata defined in the project. So here I have a metadata column enrichment round, and for every of our samples that we have, there is an assigned enrichment round. I already processed this data with the clonotyping, and I already applied the clustering analysis to cluster my antibodies by the this is VHH data to cluster my antibodies by the full length VG region. And now let's find which of our antibodies are really enriched across rounds of selection. To do so, we are going to add block, which is called Clonotype Enrichment. This is a dedicated block which is designed to track and find enriched antibodies or clusters of antibodies. So here we start with settings. And the first thing we need to select the abundance, the metric which we are going to use to track the enrichment across rounds of selection. And you already see that there are a couple of options. So first of all, we can track the antibodies and find enriched antibodies on the level of individual sequences on the individual clonotypes. Yeah, and with this option, we can use the number of reads, or since in this specific block we did redefinition of our clonotypes, we can use number of reads in our redefined clonotypes. At the same time, we can also do enrichment analysis on the level of clusters. And you see the option here. And this is a very important feature of Platforma. All the data that is generated upstream is available in the downstream blocks. So the clonotype clustering that we did previously is actually exports downstream the abundance information of each cluster across all of our samples. So that's the abundance metric we are going to use here to find enriched clusters of antibodies. Here we are selecting the enrichment round, the metadata column from samples and data that we have here, which we're going to use as a condition against which we're calculating the enrichment score, and the order of our rounds. So the enrichment goes from C0 to C1 to C2. Now there are a couple of options that are good to go with the defaults, but worth understanding. The first one is downsampling. So we can do the analysis without any downsampling, which is not good actually, because obviously we have different sequencing depths at different rounds of selection, because that's separate sequencing. Yeah, and to normalize this, we apply the downsampling, which is random sampling, which will downsample samples to the same total abundance, which is, in case of this data, is measured in the number of reads. So, and the alta or the downsampling actually means that the normalization value, the downsampling value to which the data will be downsampled, is just automatically. So this is a good option to do by default. Very good. So the other option that we have here is the clone type filtering. So in the clone type filtering, we can, well, basically it means the following. We can do the enrichment analysis either on all clonotypes, or in our case, clusters of clonotypes, meaning that even if one cluster is absent in the first round and that it appears in the second round, it will be reported as enriched clonotype or cluster. Or we can use shared clonotypes, which will perform the enrichment analysis only on the clusters that and assign the enrichment score only for the clusters that appeared in 2 rounds. So let's run the analysis. It will take some time, but pretty fast. And yes, so, well, here we have a boring output. It's just a table. So let's navigate to the, for example, frequency line plot. So this is most useful plot we're going to see. And basically it shows you the top 20 or top something clusters in their frequency or the abundance or the fraction of reads at each round of selection. And we clearly see that there are certain clusters that are enriched and have a really good enrichment score. We can also see this on the bar plot, for example. Yeah, and there is another nice visualization that you can see is a bubble plot where You see the comparison of the enrichment and then frequency of different, in our case, clusters compared to the baseline, the C0 round. So what else here is important? The stats. So you can click on stats, and here you see the general metrics, the general stats about the enrichment. So the maximum enrichment of the cluster that we have in the whole data is 14, and enrichment in this case is log₂ fold change of the frequency of the abundance of the cluster, and the default cutoff that is calculated by this block is this one. In this case, it's just 100, meaning that downstream in the down selection of the leads, the lead will be considered as enriched if its score is high, enrichment score is higher than 1.0. So how to look on this data in different ways? Well, first of all, obviously we can, for example, add the clonotype browser and see all these numbers in the table to explore our individual clonotypes. But another standard way how you're going to visualize this data before going to the down selection with the antibody lead selection log is clonotype space. So let's just, for example, add a clonotype space and visualize the entire set of our antibodies that we have in the data on the UMAP. Looking at our maximum enrichment as for example a color on the plot. So immediately on the plot we see those leads that belong to the clusters that are really enriched. Yeah, and then we can go and navigate and explore and see what's the content of those enriched candidates, what are the motifs, etc., etc. So we'll have a dedicated video about the clonotype space and how you can explore data rigorously and iterate over your data with the clonotype space. But that's it for today, and stay tuned for the next guides on antibody discovery.